whether it is heterothalism or not. Okay, that we can see, that we can differentiate. That especially by the genetic material, genetic variations, by observing the genetic variation. You know, the heterothalism is two types, two allele heterothalism and multiple allele heterothalism. If the two alleles, two sets of chromosomes variation or multiple sets of the chromosomes variation. Okay, in the multiple, either it is bipolar or tetrapolar or octapolar. These are types are happening in the. You see, first one, morphological heterothalism. Morphologically different male and female sex organs are produced to closely associated mycelia. If it is morphologically different, then only we are calling it as morphologically heterothalism. If it is associating two associated to hyphae, thallus, morphologically different. That is an example in Aclea, Aclea ambisexualis, and Bastocladia and Phytera. Majority of the heterothalism is physiological heterothalism, functional heterothalism, not the structurally. Functionally, there is a variation that identified by Blacksley first in Mucormycido. The competitive exam also they asked heterothalism first identified by or discovered by A. Blacksley. On which fungi? Mucormycido. Okay. Here, different mating types. Thale is differ. They're having the sexual incompatibility. That incompatibility because of genetic variation in the nucleus of hyphae. Okay. On that, you see, the first one is two allele heterothalism. What we call it as a two allele heterothalism? When the nuclear, both the mating type, nuclear, both the mating type differ in genetic character. Genetically, if it is differ, that is known as two allele heterothalism. You see, pair of allele representation. That means A set, capital A and small a. Both two opposites genes are at a same location, single location. You know, in the genetics you might have studied now, cis cross and trans cross, like that. Due to the dominance of A, capital A over small a is represented by plus and minus. Suppose, yeah, whatever it is. Always, you know, as per Mendel law, capital and small, internal and external characters. Bahirgata lakshanalu and antargata lakshanalu. Dominant alleles and recessive alleles. You see the multiple allele heterothalism. That means the more than two alleles, if it is deciding the sexual compatibility, that we call as multiple allele. This may be more. This may be in the same loci, same position, or maybe in the different location. Okay. First one is bipolar multiple allele. This type of heterothalism is controlled by multiple alleles at a single locus, at a same space, at a same location, different type of alleles. Suppose capital A, small a, and capital B, small b. Two different characteristic genetic material at a single location. At a single location. Suppose L, if you look one location, 1, 2, 3, 4, like that, more number of the genetic material, variation in the single locus. Okay, you can see here the four locus uh, uh, genetic place at the chromosome. You see where there is a genetic material, that locus, like that L1, L2, L3, L4. You see there is a variations happening in this. That because of this allelism. Allelism. Okay, you can see the tetrapolar multiple allele heterothalism. This is there in the Basidiomycetes fungi. You know, in the Basidiomycetes fungi, in the Basidium, you might have seen the 4 prangulas are formed. 4 basidiospores are formed because of 4 different genetic matters which are present in the same locus. That's why it formed in the 4 basidiospores. You know, in the hollow basidium, not in the progma basidium. Okay, multiple allele is a compatible factor. It may be in the single or in the 2 locations. Okay, usually it is in the 2 locations, whereas in the bipolar in the single location 2 different genes, tetrapolar in the 2 location 4 different genes. Okay, octopolar in multiple location. This also present in the Basidiomycetes fungi. Octopolar heterothalism. This is controlled by 3 factors, A, B, and C. 3 different factors on the loci which are affecting for the octopolar heterothalism. Usually, in the lower fungi, zygomycetes, whenever the zygos parenchyma formation is taking place, there 2 opposite strain thallus only is mating. I already told you 2 opposite strain gametangiums comes together and mate. Cell wall dilution is taking place and forming the zygospore. Whenever 2 opposite strains, plus and minus, that is there in the usually rhizopus, mucor, aspergillus. Okay, you see in the mucor, you know, bread mold, black mold, it is happening. You see asexual spores by chlamydospores and idea. There it is what is happening. 2 opposite strain thallus only is being made. You see 2 opposite strain thallus only, plus sign minus of strain. There only it is plasmogamy and karyogamy, zygote formation, zygospores, and again taking the reduction division and releasing the haploid spores. The haploid spores are released by the rupturing of sporangium. The meiospores, reduction spores, haploid spores are releasing. You see, mature gametophyte again forming, but again the reproduction plasmogamy, karyogamy, and meiosis is taking place in between the 2 opposite strains only, not in the same strain thallus. That is heterothalism. In higher fungi, especially in ascomycetes, There is a much greater variation is there in the higher fungi. The hormonal variation also is there in the higher fungi, in the heterothalism. You see here in the Pragmobasidium, that means in the, you might have studied the Puccinia graminestridaceae. We trust it is a macrocyclic life cycle. It are having the two hosts, primary host and secondary host. There it is happening. You see, Pragmobasidiums, Teleospore, Probasidiums are forming, that are mating and forming the esiospores, pycnospores. It is happening because there is a variation in the sexual hormone. Here you can see in Ascobolus sexual cycle. See, Ascogonium and Anthridium. Opposite strain, Ascogonium anthridium only is taking fusion, and dichordization is taking place. With the dichordization, again it forming the diploid, that again taking the reduction division, forming the eight spore structure, ascus. These all are germinating and forming the new. You see, the mating is happening in between the two opposite states. There is a compatibility is there in between the two opposite states, Ascogonium and Anthridium. That is heterothalism. Very, very important in the competitive exam point of view. Your theory examination means having the different strain of the genes or different genes with the same type of the either plus strain or minus strain type of the genes in the single thallus. Not the different types are presenting in the single thallus. That we are calling as heterothalism, or also calling it as monoecious. Okay. And the types of heterothalism also we have discussed. Morphological heterothalism, physiological heterothalism. The physiological heterothalism again, 2 allele and multiple allele heterothalism. The multiple allele heterothalism again tetrafloid, octafloid types we have studied. You see, bipolar, tetrapolar, and octapolar. Okay, bipolar in the same locus, tetrapolar in the two different locus, octapolar in three different loci. In the lower fungi, especially Muca, Rhizopus, and higher fungi, especially Aspergillus. Okay, this happening in. Okay, and the next foremost important is Heterokaryosis. This Heterokaryosis means two different nuclei are present in the same thallus. If the two genetically different nuclei is present in the individual thallus, in the same thallus, that is called as heterokaryosis. Karyo means nucleus, hetero means different. Different nucleus are coming together with each one, and the genetical exchange is taking place. Okay, that is called as heterokaryosis. Okay, the two sets of chromosome lie in the separate nuclei, sharing the same cytoplasm. That means dichordization, two nuclei within the same cell. Two opposite strain nuclei, not the same strain nuclei. Okay, You see, it is dominant the diploid. You see, hetero dissimilar karyosimous nuclei. Coexistence of genetically different nuclei within the same phallus, within the single cell. Coexisting of two different genetical nuclei in the same cell, in the single cell, is called as heterokaryosis. Heterokaryosis first discovered by Hansen and Smith. Heterothalism first discovered by A. Blacksley in Mucormycidae. Heterokaryosis first discovered by Hansen and Smith in Botany cinerea. This plays major role in variability and sexuality in haploid fungi. Okay, it is one of the prerequisites where there is a two different genetically different nuclei is present in the single cell. That cell will leads to the parasexual cycle instead of normal sexual reproduction. Okay, like heterozygosis. Heterokaryosis also one of them. You see, the formation of heterokaryosis is taking place by three methods. How many? By three methods. First one is mutation. Normally, by the mutation only, genetically variation nuclei may comes together at one place. Usually, it does not happen, but it may be diffused. But whenever the mutation is taking place, and second one is anastomosis. Anastomosis means fusion between two different genetically different hyphae. Plus 10 minus 10 hyphae fusion. That is heterothalism. That heterothalism is leading to heterokaryosis. Heterokaryosis is leading to parasexuality. There is an interconnecting link between the heterothalism, heterokaryosis, and parasexuality. Okay, and then inclusion of dissimilar nuclei. Where it is happening? In the heterothalic fungi, whenever the two opposite stain thalli come together, two opposite stain nuclei, two genetically different nuclei are mating. And after some time, it taking the reduction division, meiosis. So that at that time, it is exchanging their genetic material. Okay. This mutation, high frequency mutation, causing some time for this heterokaryosis. Variability of the sources. Especially Penicillium, Aspergillus, Verticillium, Fusarium. We can't for your condition. Mutation frequently happen in this group of fungi. See, because sometimes this heterokaryosis, they demonstrated that as a new trace instead of same fungi, but because of genetical modification, we face it as a new stream. Why? Genetical variation is there. There is not that. You see, here, sometimes single spore of Fusarium frequently get mutations and producing various types of the genetic material. That we are calling it as dual phenomenon. And anastomosis, that we are studied heterothalism. two different stain thallus. That's happening. Two different genetically different nuclei are mating. Genetical modification, high frequency, high potentiality will come, will gather. Interspecific anastomosis is also happening between Botrytis cinerea and Botrytis reginae, not only within the same species. Yeah, nuclear migration from point of fusion to other areas. So that it is happening where there is a initiation in the point of fusion and that nuclei dividing in from the both the parent hyphae. So that some of the genetic material is going to side cell, adjacent cell, that is going on. See how the fusion of hyphae, dichordization, secondary mycelium formation in the basidiomyces especially. By clamp connections, outer ring road, bridge formation. These are these are forming the bridge formation, clamp connection, exchanging the nucleus from one cell to other cell instead of going directly. Outer ring road very fast, and inclusion of dissimilar nuclei in spore after meiosis. What happening after meiosis taking place in the two different nuclei? Whatever the spores are formed, that two opposite two nuclei spores are mating, and again it is forming the diploid. Dikaryotic will become diploid. Frequently it is happening so that multinucleate spores, diploid spores and haploid spores, both the spores are presenting in the single thallus, single cell. That is happening. Haploid spore and diploid spore both are happening, sharing common cytoplasm. That is causing for the heterokaryotic. You see, there is a interlink between the heterothalism, heterokaryosis, parasexuality. Here you see the parasexuality life cycle. What is happening? Instead of normal sexual reproduction, instead of continuous sequence of plasmogamy, karyogamy, and meiosis, these are not taking in the specific area, in the specific time, but it is not happening. Exchange of the genetic material is happening. That is nothing but parasexual life cycle. It is looking like sexual reproduction, but it is not exact sexual reproduction.